Over the past few years, I keep coming back to this question of embalmers clots, not because I think it's controversial, but because I think it has really important clinical ramifications. What people don't quite get is that embalmers clots, what embalmers are seeing in terms of when they're doing the post-death clearance of the vascular system, flushing out all the clots, they have been coming across really strange fibrous white clots that they haven't seen before the pandemic occurring. And many people perceive this as just a conspiracy theory. My view is very simple. The embalmers are likely to be seeing this because they are seeing severe cases of what occurred and those people died. And that's why you're having these embalmers clots. But you just have to do the extrapolation and think about the implications. Suppose the clots are not as big to cause someone to die. What if it just causes them to have clots on their lungs, clots in the brain, clots in the heart? These are the points that are very relevant because if they are of similar characteristics, very fibrous, very difficult to break down, it gives little opportunity for the body to handle them. And that's the clinical context. And so, therefore, we have to keep looking until we find answers. Part of the reason I'm coming back to it again is because of this paper, Persistent Epigenetic Memory of SARS-CoV-2 mRNA Vaccination in Monocyte-Derived Macrophages. This was a very important paper, in my view, because it showed me and gave me an answer that I had been looking for a long time. I just didn't think it would come from that angle. And that was to do with how macrophages, especially post severe COVID 19, because in severe COVID 19, macrophages are central to causing the problems. And so, in the longer term symptoms, we knew that macrophages were involved as well, but were they central? This paper definitely positions it as such. And if you want to be able to see that, make sure you go on the link to the Substack, DNA Reprogrammed, how this hijacks the immune system. And you'll understand the basics about what I was talking about with regards to histones and looking at some of the links in the paper and so on. That's the advantage, by the way, when you when you come on Substack, is that if you see the video, not only do you see the video, but you also get a full article with the links explaining oftentimes what we're talking about. So it's quite useful for some people. If you just want information about what was spoken about in the past with embalmers clots, you can also get that in the link below in the description. I did this presentation sometime last year based on all the information and effectively came up with white clot syndrome. And I did a whole overview looking at the protein constituents of the clots, hemoglobin concentration, links to autoimmune heparin-induced thrombocytopenia. You got the questions and answers, you get the full scientific publication, and even observations from a vascular surgeon. Believe it or not, that whole presentation is free. So before it changes, take a look at it. It's very valuable. And so coming back, I have been looking at these concepts of macrophage activation. I'm going to go through a short presentation, which again will be on Substack now, where it takes you through a little bit more of the scientific analysis as to what I'm looking for with regards to these embalmers clots. And remember, even though I say embalmers clots, what I mean is unusual fibrous clots that can occur in the living as well as in the dead. So here are some of the concepts that are relevant. Starting here, this is what we're talking about when we say an embalmer's clot. This here is Richard Hirschman. He had pulled one out. Look how long this is. It is 19 inches long. He put it in formalin so that it's stored. It's almost completely white all the way through. we have never really seen anything like this. As I said, the only closest approximation is white clot syndrome, which is heparin-induced thrombocytopenia. So for those people who think that this is a conspiracy, it is definitely happening. We just don't fully understand all the mechanisms. Recently, I spoke about the autofluorescence that we noted, or that was noted by a scientific team looking under UV lights, which suggests some very unusual patterns with these clots. And why it's important, and we thank Tom Haviland for continually raising awareness of this. And he did this study where he reached out to embalmers all across the world. 301 answered, and of that, 83% of them observed large whitish fibrous structures or clots or structures in the corpses that they embalmed in 2024. This is not since 2021. In the year of 2024, 83% of them still are seeing them, up from 73% in the year of 2023. This is huge. And as I said at the time, when we spoke about it in about December, January, this was really, really important. And similarly, when he asked them again, the embalmers, question 4 was, did you see the large whitish fibrous structures in a higher percentage of corpses in the year 2024 than in 2023? 59, so less, 155 about the same, more for 47, so none 40. This is quite significant. So it's about the same for about 50%. More clots, just under 20%. This is quite significant information. And as I said, this is why I keep coming back to it, because it is not acceptable to not fully understand this. It is a pattern that is almost unique to embalmers because they are the only ones who will flush clots out of the blood system. Recently, I spoke with a doctor who also does postmortems, and he also found these strange, unusual clots. So this is being seen not just by embalmers, but on postmortem. And critically, we know that similar clots are also found in catheter labs being pulled out from people who are seriously ill. So this is important stuff. Here is one of the analysis from a female teenager who had one of the clots, and this was then done in a lab. And this is the bit that is coming into the science of it. Hemoglobin subunit beta is red, fibrinogen is blue, myeloperoxidase is green. So these are the main constituents in her. It was slightly different for other people, but the main concentration was hemoglobin. And as I said at the time, I was focused on the fact that this could have been a mirror of white clot syndrome, heparin-induced thrombocytopenia. Well, the research that I mentioned about the epigenetic changes in macrophages is now tying together what I think is a very important piece of the puzzle with regards to the science. So this is complex stuff, but let's work through it. The first thing you have to understand is something called netosis, neutrophil extracellular traps. Okay? And this is what it is here you have a neutrophil, and you can see that the cell wall has burst. The nuclear material is being released, and it has all these strands of chromatin. And this is what you would call an extracellular trap. It's very sticky, and it sticks onto bacteria or viruses and so on. And so it's a unique immune response where neutrophils will release these web-like structures, DNA, histones, antimicrobial enzymes to trap and kill pathogens. It can be triggered by infection, inflammation, or even stress. Critically, these nets are prothrombotic. That means they cause clots, and they contribute to microvascular clotting and organ damage and autoimmunity. So this was a critical part of what happened in severe COVID. But it seems as though it is still occurring in the context of reinfections. And this is what I am thinking is the main driver of these very unusual clots. But here is now where we take the next step up, and this is linked to the epigenetic changes in macrophages. So again, let's go back to the science. What we have here is a macrophage. They are very unique and they're very key innate immune cells. And they critically do phagocytosis. That means they literally form a bulb around something, swallow it up, pull it inside, and then destroy it. They can present antigens and they oftentimes produce a lot of cytokines. And they're usually in the either the pro-inflammatory state M1 or anti-inflammatory state M2. The fact that what we have learned from the epigenetic changes suggests that these are being driven in the pro-inflammatory state. And that's part of what I think is the problem. And in my mind, when I looked at it, this is what it looked like. This activated macrophage here, you can see it. Inside here's the nucleus. It has all these little pseudopods here, which grab things. And it's producing cytokines all the time because it's activated. Now, you have to remember that they realize that the mRNA keeps these cells locked in an activated position. And for them, when they were doing the study, they said that's great because it means it's going to target the necessary pathogens and it stays activated for up to 6 months, maybe longer. But there's an important difference with regards to these specific things is that when we look at them and we go now back to the paper of these epigenetic changes, this is what happens when you look inside the genes. This is what I want you to concentrate on. What they had found, these are the unvaccinated. Now remember, unvaccinated means natural infection. I have always used this as a template to understand disease. Let's make this a little bit bigger. And the reason why I use that is because it tells us whether or not this is how it should work. The genes that are expressed overlapping, you can see here, if you add only 50 were unique to the unvaccinated, 268 overlapped both the unvaccinated and the vaccinated. 2,519 genes were triggered in the vaccinated cohort. This to me is a big problem. I don't think this is a good thing. Whatever the paper may tell you, they think it's great. I don't think it's great because I am understanding that the implications with regards to what happens in terms of long-term immune mechanisms is very, very difficult to understand. Here is what I think then is happening with these macrophages, which are becoming hyperactivated. Guess what? They also do traps, just like neutrophils. It's what we call macrophage extracellular traps. And this is what it could look like. This here is your macrophage. It's activated and it releases these nets. The interesting thing about macrophages, unlike neutrophils, which die when they do it, the macrophages don't. They almost like a spider's web can release multiple of these these traps to trap all kinds of things, whether it's bacteria and so on. But in the terms of autoimmunity, this now becomes a huge problem. And this is now where it's going to tie in to what I said about Embalmers clots. So here is the summary of the idea. In an immune-primed individual, and truthfully, this is most likely going to be in the vaccine immune-primed individual, on re-exposure to the virus in some people, not in everyone, we can then have a hyperactivation of the immune system. And this hyperactivation of the immune system will cause macrophages, which are already hyperactivated because of the vaccines, to start releasing traps. They will then, because of the cytokine release, trigger neutrophils to do the same thing. And the two together will then be releasing nets to trap all kinds of proteins in the bloodstream into one huge mush. And this is how I envisioned it. This was my creation, and I hope it makes sense to you. But this is how I saw it. Here we have one macrophage releasing an extracellular trap, another macrophage doing the same. And as it releases them, it starts to trap immunoglobulins that were targeting spike protein, and it's trapping fibrinogen, it's trapping hemoglobin. And if you multiply this by thousands, suddenly this whole conglomerate of protein enzymes all trapped together can then cause very unusual, very difficult to dissolve clots. And this here is the center of the netosis. So the way to imagine it, because these can do multiple nets, imagine them throwing them in multiple directions with each other, trapping all the proteins together to then form a very solid, whitish, fibrous protein that we have not seen before. And this is my prediction as to what is happening with regards to these unusual clots. It's important for us to reflect on what the science is going to tell us. As I said, science should never have ignored what embalmers were observing for almost 4 years now. I think that's completely, there is no good explanation for it. Whatever anybody may have thought about a conspiracy, if it's real, if it's occurring, and it has not occurred before, it is almost negligent not to investigate. That is the only logical thing I can say. And so as we continue to try and find the mechanisms, because the speed of these formations, they seem to occur quite quickly because they don't seem to be just casually lying around. So some people say, well, do I have them in me? If you are well, it's pretty unlikely. But if someone gets sick, it's quite possible that these kinds of immune-mediated clots can form very quickly, within hours, within a few days, to cause someone to then get very sick. And this is why I think that we must try and understand this, because at the end of the spectrum is embalmers clots, huge clots. But what about in the middle, where we are having small clots being formed? What diseases will this occur? Multiple clots, infarcting multiple organs, and no clear explanation. That is the pattern that we could see in the near future if we don't find ways to clarify what is going on. Thank you for listening. And again, if you haven't seen the full video, please make sure you join on Substack. You'll get an article associated with it, and we'll continue to bring you the latest updates on the science. Have a good evening.